Good morning to our lecturer and fellow classmates. Today, our group is presenting on a significant challenge in the global food industry, which is the identification of forgery in meatballs using pork. This presentation is part of our assessment for STKM 3222, Management of Halal Food. As the first speaker, I will provide the background of this issue, explaining why the specific food product is highly vulnerable to fraud and how it affects the halal status for Muslim consumers. To understand the background of this issue, we must look at the nature of the meatballs. They are what we call comminuted meat products. This means that the meat is finely ground and mixed with binders and spice until it becomes a uniform paste. Because of the physical structure of the meat is completely changed during processing, it creates a detection gap. A consumer can no longer identify if the meat is beef or pork just by looking at the texture or tasting the cooked product. This made meatballs a high-risk target for manufacturers who wish to hide prohibited ingredient. This leads us to the definition of forgery. This is not an accident, it is a deliberate act of adulteration where manufacturers substitute beef with pork to reduce costs and increase profit. From a halal management perspective, this is grave violation. First, the presence of any pork, even if trace amount, renders the product is strictly haram, and as it is a najas al-muhalazah. Second, it violates the principle of halalan thayyiban, which is require food to be wholesome and the trade to be honest. Understanding this background is essential as we move into the specific law and identification method. I will now pass the floor to the second member to discuss the issue involved in more detail.
Now that we understand the background of the meatball industry, we can move on to the key issues involved in this meatball forgery. One of the major issues related to meatball forgery is economic fraud, where pork is intentionally added because it is cheaper than beef or chicken. This unethical practice allows producers to reduce production costs while selling the product as halal, which can mislead consumers. Scientific evidence shows that this issue is real. A study published in Chiang Mai Journal of Science used DNA-based analysis to investigate processed meat products. The sample were analyzed using polymerase chain reaction PCR techniques. This confirmed the presence of porcine DNA in the meatball product claimed to be beef. This finding provides strong evidence that meatball forgery is possible and can go unnoticed without scientific testing. The detection of unclad pork in meatballs raised serious concern, especially for Muslim consumers. These products were sold without proper disclosure, leading to consumer deception and also violation of halal requirements. Beyond religious implication, this practice also affects consumer trust and food integrity. Overall, this case demonstrates that meatball adulteration using pork is a real and documented issue. It highlights the importance of strict halal monitoring, accurate labeling, and also the application of reliable scientific detection methods in order to protect consumers and also ensure food safety.
For this part, I will explain the Islamic law related to the pork consumption. In Islam, pork or khinzir is clearly prohibited and classified as haram. This prohibition is explicitly stated in the Quran, particularly in Surah Al-Baqarah verse 173, which mentioned that Allah forbids the consumption of carrot, blood, and the flesh of swine. This verse serves as the main legal basis for the prohibition of pork in Islamic dietary law. Mufti have affirmed that pork and all pork-based products are haram in all forms, and any food containing pork cannot be considered halal. Additionally, Islam strictly prohibits the food deception or tadlis. Therefore, sellers are responsible for ensuring truthful labeling and maintaining halal integrity. This slide highlights the issue of meatball forgery involving pork. When the pork is added into the meatball and the product is sold as halal, it clearly violates Quranic law. This action also contradicts Fatwa ruling, which states that any food containing pork cannot be considered halal. More importantly, this practice involves deception of Muslim consumers as they mislead into consuming food that is religiously forbidden. In conclusion, meatball forgery using pork is haram and unlawful in Islam. It threatens halal integrity and consumer trust, which is why scientific detection method.
So next, I will present the identification method for pork forgery in meatballs, focusing on the what, the specific biological and the chemical targets we look for in the lab. Our first and most reliable target is porcine DNA. Specifically, we look for mitochondria DNA, mtDNA, such as the cytochrome B gene. So why this target? So firstly, mtDNA exists in thousands of copies per cell, making it much easier to detect compared to regular nuclear DNA. Second, it has high stability toward heat. This is crucial for meatballs because even after they are boiled at high temperatures, these small DNA fragments remain intact for us to detect. This ensures we can find the genetic fingerprint of a pig. So next, we target specific proteins. These are proteins found in a pig muscle tissue that differ chemically from beef or chicken. So why this target? Because we specifically look for myofibrillar proteins or heat-stable antigens. While many proteins break down during cooking, these specific antigens remain detectable. This target is very useful for rapid screening and detecting pork in a raw or lightly processed meat using quick test kits. So our final target is porcine lipids or lard. We target at the unique chemical profile of triacylglycerols or TAGs. So why this target? Because the arrangement of fatty acid on the glycerol backbone in pig is very unique. By targeting these molecules, we can detect if pork fat was added to the meatball mixture to save costs even if the meat used was originally halal.
In order to identify whether pork has been fraudulently added to meatball samples, there are several scientific methods can be used. The first method is real-time polymerase chain reaction qPCR, which is used to detect pig DNA. First, DNA will be extracted from the meatball sample. This DNA contains genetic material from all the meat that used to make this product. Then, pig-specific primers will be added. These primers are short DNA sequences that only recognize and bind to pig DNA. During the PCR process, if pig DNA is present, then it will be amplified, meaning that many copies of pig DNA will be produced. At the same time, a fluorescent dye will also be released. As more pig DNA produced, the fluorescence increases. This produces a rising curve which indicates the presence of pig DNA in the meatball sample. The second method is ELISA, which is used to detect pig-specific proteins such as porcine myoglobin or immunoglobulin G. First, protein will be extracted from the meatball sample. Then, specific antibody that recognize this protein will be added. If pig protein is present, these antibodies will bind to the pig proteins. After that, an enzyme called horseradish peroxidase or HRP will be added. This enzyme will attach to the antibody-protein complex. Next, a color-forming chemical called TMB will be added. Finally, sulfuric acid will be added, which changes the color to yellow. If yellow color appears, it indicates the presence of pig DNA in the meatball sample. The third method is Fourier transform infrared spectroscopy or FTIR, which is used to detect pig lipids. First, the lipid will be extracted from the meatball sample. Then, the FTIR instrument will scan the lipids using the infrared light. Then, this produces a spectral fingerprint that represents the chemical structure of the lipids. The spectrum is then compared with reference spectrum of the pork lipids. If the peaks and pattern match the reference pork lipid, then it indicates the presence of pig lipid in the meatball.
Based on Islamic law, pork or khinzir is clearly prohibited and classified as haram. It is also considered najas al-mughalazah, which means it is a severe form of impurity. Therefore, any food that contains pork, even in a small amount, cannot be considered halal in any form. In the case of meatball forgery, the act of adding pork into beef meatball directly violates Quranic law. This practice also involves food deception or tadlis because the product is sold as halal while containing haram ingredients. Islam strongly emphasizes honesty and transparency in trade. Thus, this deception contradicts Islamic ethical principle and violates the concept of halal and thayyiban, which require food to be lawful, safe, and pure. This issue does not only affect religious compliance, but also has wider implications. Muslim consumers rely heavily on halal labels when choosing food. When pork is secretly added, consumers are misled and their trust in the halal food industry is broken. This practice threatens the integrity of halal certification, undermines food safety standards, and causes ethical, religious, and social harm. In conclusion, meatball forgery using pork is clearly haram and unlawful in Islam. The issue involved two major violations: the use of haram and najis ingredients and intentional deception of consumers. Scientific detection methods such as DNA, protein and lipid analysis are therefore essential to ensure halal authenticity. Strict monitoring and enforcement are needed to protect Muslim consumers and maintain confidence in the halal food industry. Halal is not only about the ingredient used, but also about honesty and responsibility in food production. Thank you for listening. We now welcome any question.